Hi, everyone, and welcome to the Genetics Podcast. I'm really excited to be here today with Dr. Nicky Whiffin, who is a group leader at the Wellcome Centre for Human Genetics in Oxford. Nicky leads the computational rare disease genomics group, which uses computational approaches to interpret the role of genetic variants in rare diseases. Nicky, it's so great to have you today. Thanks so much for joining me.
It's great to be here. Thank you.
I'd love maybe if we could just start, for people who aren't familiar with your work, to have a little bit of an overview of what you and your research group are working on and how you got to work on rare disease genomics in the first place.
Yeah, definitely. So our team looks at trying to find diagnoses for patients with rare disease. So traditionally, our approach is when a patient turns up in a clinic, they have genetic testing done. And traditionally, approach is to only look at a very small proportion of the genome, around 1.5%, that directly encodes the proteins that go and do work around our body. With that approach, we're relatively successful. In some diseases, we can diagnose genetically kind of 90% of patients, but in others, this is severely limited. So it's more like 20 to 30%. So we're clearly missing something, and we try and figure out a bit more about what we're missing. So we focus on variants that are outside of this protein coding sequence, but our main interests are what we kind of term near coding, which are regulatory regions that are directly adjacent to these protein coding sequences that have incredibly important roles in regulating the amount of proteins that are produced. Our favorite ones are untranslated regions, or UTRs, that are directly up and downstream of the protein coding sequence. And they do all sorts of roles like regulating the stability of RNA. So that's the kind of precursor before protein, so between DNA and proteins. And that RNA kind of persists in the cell and gets translated into protein. But these untranslated regions regulate how long that RNA persists and the rate at which it is translated into proteins. They have incredibly important regulatory roles. And we're interested in finding the subset of variants within those regions that actually can have severely detrimental effects and lead to disease.
So what, in terms of kind of scale or real estate, you mentioned about 1.5% of the genome is coding. What percentage then would be in these near coding regions that you all are looking at roughly?
Yeah, so the thing that people don't realize is that UTRs are roughly equivalent to the size of the protein coding sequence for each gene. The majority of that is the 3' end, so they're kind of downstream gene end, and they're kind of smaller, the 5' ends are upstream is much smaller, so that they can be as small as, say, I think the average is around 200 base pairs, but they can be much longer. And actually, we're finding a lot in our research at the moment that genes that are important in their dosage, so loss of function intolerant genes, tend to have much longer, more complicated UTRs that have more regulation in them to tightly regulate the translation of those important proteins.
So how much people listening may be familiar with the triplet code? We've got a pretty good, although not perfect, understanding of what a change to a protein coding section of DNA does to the ultimate protein. How much do we know about the code in these near coding regions, and where are we at a few years ago? Where are we at today, and where do we need to be?
We know very little, still know very little. I don't think we've made a huge amount of progress in that code, and we need to do a lot of research on that. One of the things that makes it particularly tricky is when you look at a regulatory element, it acts as a whole. So if you're looking at the triplet code and predicting the effect of a variant on the protein, you can predict that a single variant will change a single amino acid. Whereas in, say, a UTR, a single variant might shift a structure which affects the regulation as a whole, or it might have actually no impact but look like variants that have a big impact if they're in a different portion of the UTR. So it makes this incredibly difficult, and you have to look at things on like a UTR by UTR basis.
And I guess every gene's got different size UTRs, probably different structures within it. Are there supersets or groups where you can start to say, are there any patterns that you can start to build up, or is it really starting from a blank slate almost, where we know these things have a function but we're trying to understand the language, so to speak, now?
Yeah, so the UTRs are incredibly variable across different gene sets, not just in terms of length, but also the makeup of different regulatory elements within them. Some of the variants we've most recently studied are ones that create upstream start codons. So they create just an ATG within the UTR. And that kind of causes the ribosome to pause and decide whether to translate early, and can kind of disrupt the translation of the downstream proteins. These are variants that we know can be very impactful. But again, it's very dependent on where they occur and in what context they occur. So it might be that they occur, but they occur downstream of the start that's already being used, which means that they don't actually get seen by a scanning ribosome that does the translating. Or it might be that they are created into a context that actually isn't favorable to the ribosome, so it completely ignores it and keeps scanning and actually makes it to the downstream coding sequence. So there's a lot more complexity in just creating an ATG. It's what is that context is that created into?
What are you starting to learn? I think one of the interesting approaches that you and your group take is you look at cases where something has gone wrong with one of these near coding regions. So a child or an adult has a rare disease that is likely or potentially the result of one of these changes. And what you can do from this, I think, is start to understand by what happens when we break this element. We can learn a little bit more about the function of the element itself. Maybe you could talk a little bit about how that process works, what you've learned so far, because sometimes there's a huge amount of value in a small number of cases where you can really have a breakthrough, almost, where you understand, by understanding what happens when it goes wrong, we can learn something fundamental about how these mysterious various parts of the genome work as a whole.
Yes, one of the things I find most fun about working in this area is it's not just about finding the diagnoses, it's also about finding something new about fundamental biology and kind of teaching. So the upstream start codons that exist in 5' are a natural occurrence, actually. So they create what we call upstream open reading frames, or UOF. And these naturally occur, or they're predicted to occur, in around half of all known protein coding genes. And just kind of as an example of how little we know about these elements, is I had a talk at the American Society of Human Genetics conference a few years ago. And it was great fun to stand in front of a crowd of human geneticists and tell them that these elements exist in their genes they've been focusing on for kind of decades, that they've never even heard of before, but they're regulatory elements that are very, very common. And we can kind of, by looking at the variation and the way that leads to disease, we can look at when these elements are more, I can't think of the word, not the elements themselves being more deleterious, where they have more of an impact on protein translation. So they're kind of cis-regulatory elements that downregulate protein translation by disrupting this ribosome scanning. But if you find a variant in a patient that's deleterious, you can see, okay, that's obviously a very high impact variant that's having a really big effect on downstream protein translation. But then you can look in kind of population controls and say, well, these genes or these particular types of variants don't seem to have such an impact on regulation. And from those genetic variants, you can actually learn when these elements are more regulative and when they are kind of not having such a big impact.
And what's the estimated impact on patients and diagnoses? I know you worked recently with Jamie Ellingford and a number of other scientists to look at clinical interpretation of variants in non-coding regions. And as you know, I've been also interested in this, and it's what I did a lot of my research in. And it's still an area of clinical practice that's really very challenging. Where are we at today? How many patients do you estimate may have diagnoses that are not being made in most labs around the world because we're not looking at these parts of the genome?
People ask me this question all the time, and it's a number that I really, really want to have, like to pull out of my back pocket. Sadly, we don't have that yet. One of the problems is kind of, it's hard to do these analyses to get a real number on that because there's also so much noise in the non-coding space. I mean, a lot of work you did during your PhD had the conclusion that it's individual bases that have an impact. It's not entire regulatory elements. So you've got to find ways of filtering the variants that you only got the ones that have a large impact. And if you don't do that properly, you end up with a lot of noise. And one of the ways you were looking at this was to look at de novo variants, so ones that occur only in a child that has unaffected parents, so they have a higher prior of being disease-causing. And we have some work we're doing in the Genomics England 100,000 Genomes project at the moment, where a superstar postdoc in the team, Alex Geary, is working to try and look at both de novo and inherited variants, and using, we think, hopefully some clever approaches to try and finally put a number on that. What proportion of patients have a likely diagnostic UTR variant or promoter variant or other kind of regulatory variant? But at the moment, we don't really know what we're expecting to find. And the most progress in this field has come from single gene disorders. So patients where the clinician can say that patient has a variant in that gene. But in a lot of the kind of developmental disorder space, for example, there are so many genes that could be the cause. It's much more heterogeneous, which means that you can't really pinpoint exactly which gene you're expecting to find a variant in. But there's some kind of nice poster child genes. So we did some work recently with Caroline Wright and the Deciphering Developmental Disorders team at the Sanger, and we kind of found this gene called MEF2C, where actually we find that 25% of patients in DDD with a MEF2C variant have one in the non-coding region. So for some genes, this non-coding variants are incredibly important, but for others, you can diagnose, say, 90+% of patients in the coding sequence. So it's very, very gene, region, yes, everything dependent. So it's very hard to put a number on that.
Do you have a sense of whether that gene MEF2C is more of an exception or more of a rule? Do you think that if, and I'm asking you to speculate here, it'd be great if you had the data, but yeah, curious whether that is an outlier or not.
As high as 25%, I think it's definitely more of an exception than a rule. I think most genes it's going to be lower than that, but it's not going to be the only one. And it's some kind of combination. I think the main factor for this is probably because it's very dosage sensitive. So even the variants that in some genes we wouldn't think would ever be that deleterious, in MEF2C, it seems that they can cause a severe developmental phenotype. So that also factors in it. And also just the fact that we had coverage in the sequencing data to actually be able to profile the variants in that region, which is obviously very important to what we can detect, because DDD, as you know, is based on exome sequencing data, so only capturing the protein coding regions. However, I was talking to some people yesterday how that's a bit of a misnomer, because whole exome sequencing does not sequence the whole exome. Right. There are lots of exons that are non-coding, so whole exome sequencing is a bit of a misleading name for the technology. When
you and I were both earlier in our careers, there was and honestly the term's still used, but this term of junk DNA, of the non-coding genome being 98% or 80%, depending who you ask, junk. I think we're learning that that's definitely not the case, but I'm curious what piqued your interest in the first place of studying something that a lot of the field said was, basically the actions going on in the protein coding genes. Sure, there's some important stuff, but what made you interested in focusing on the part that nobody else was actually all that interested in?
Yeah, I think the junk DNA term just kind of highlights how we'd rather discount something that we don't understand than to realize we have to put a lot of effort into trying to understand it. My PhD was actually on doing a lot of genome-wide association studies in colorectal cancer, and obviously a lot of GWAS hits are found in the non-coding regions. I found that kind of quite unsatisfying because I wanted my research to have more of an impact on patients, which is why I kind of shifted away from GWAS and into the kind of rare variant, rare disease fields. But I think it's part of that that's combined my interest and also just kind of frustration a bit in the fact that we ignore. So we've spent a lot of effort in trying to find the genes that are involved in certain diseases, but then we ignore half of the gene. So we actually only focus on the bits that code for protein, but we actually know that these genes are important in this disease. So we should be looking at the regulatory elements for those genes as well. But the fact that we don't is mainly because we just don't know how. So if I can do a little bit towards trying to teach us how and make some difference in that way, then I think that's a very rewarding thing to do. So that's kind of how I got to this space. But
yeah, and I think you all are doing really important work, and I think Genomics England is doing important work here, where they're sitting in between clinical practice and research, because I think one of the challenges has always been, if you're running a clinical lab and the question is, do I charge somebody $1,000 to do a whole genome or $300 to do a whole exome, and we only know about the whole exome, so that's all we can interpret, then it's really clear there's not a justification to insurers or National Health Service or whatever to say we're going to sequence all this stuff that we really don't know anything about and we can't report on. But you end up in this catch-22 where if you're not sequencing it, then you don't know what we're missing. And so most of the clinical sequencing around the world is either single gene, multi-gene panels, or starting to be exomes. But what I think Genomics England and a few others have done to really start to break this cycle is say we're going to do whole genomes because we don't know what we don't know. And by forcing the issue, we can actually really figure out, is this going to be worthwhile in the long run? And I think starting to chip away at that and say, how many diagnoses can we get out of this in-depth sequencing? And I think when you do the math, especially as the cost of sequencing comes down, it's going to start to make sense very soon, if it doesn't already, that we should just simply be whole genome sequencing. And everybody, right? Even if you can boost diagnoses by 5 or 10%, the impact of that is so enormous on the patients and the costs that I imagine the numbers start to add up. Is that your view on it as well?
Yeah, I kind of think there might still be some diseases where panel testing makes sense, but it may well be expanded panel testing. And if we have a set of standardized regulatory regions that also should be included within that panel, then it might make sense. And if we can diagnose 99% of patients using an extended panel, then there is no reason to go down whole genome sequencing. One time where it really can make an impact is where we have these long diagnostic odysseys for these patients. And if you're going in and you're doing karyotyping, then you're doing panel testing, then you're doing exome, then you're finally getting to genome, that's a long time for all of those processes to work out. And that's just adding to the kind of uncertainty for these patients. Whereas if we go in with whole genome, we can still focus our search first on kind of expand it as we go along with the kind of more likely candidates first. But the data is already there. And actually in the long term, you're saving money and you're also kind of saving a lot of kind of time, both for the patient and also for the kind of scientists doing the work.
Have you all looked at other kinds of sequencing technologies, long read sequencing or anything like that? And is there value to something beyond the current short read sequencing that we're all more or less used to working with?
I think there's definite value, but I've not looked at any long read data. One of the kind of really interesting things is being able to do phasing. So not necessarily having to have access to parents to be able to know which parent something was diagnosed, to be able to find combined heterozygous variants and prove
maybe you could explain. Not everybody, for those who don't understand phasing, I think it'd be a really important concept. Maybe you could jump in and explain what that is.
Yeah, sorry, that was a big bit of jargon, but I just
no, it's great. It's really good. I don't think we've had an episode where we've really talked too much about it, but I think it's such an important point, especially in rare disease.
Yeah, so this is where you inherit one copy of each chromosome from each parent, and it's difficult when you're only sequencing very short sections of that to figure out whether a variant that is 500 base pairs away from a different variant came from the same parent or came from a different parent. So what phasing is trying to do is trying to predict kind of computationally which variants arose on the same, like what we call haplotype, but from the same parent, and which ones came from a different parent. This is important when we're looking at a recessive disease where we need, say, both copies of a gene to be knocked out. And if you see two variants you would expect to knock out a gene on the same haplotype, then that's only going to knock out that single copy. Whereas if they occur on the different haplotypes, they've been inherited from different parents, then you get both copies knocked out. So one of the ways we can find this out is by sequencing the parents, and you can see which parent had which variant. But that's obviously adds to the cost. You're doing 3 times the amount of sequencing. Whereas if you can do phasing, you can find, or long read sequencing, you can find this out just from the patient themselves.
Having access to phased data and understanding not just the child's genome, but both parents, I imagine is really important for what you do, right? Do you have a sense of how much that improves the diagnostic rate, or how important it is to actually have that complete family information versus just data from an individual?
Yeah, I can't remember the numbers that go with this, but there's been lots of studies that have shown how the diagnostic rate is far higher if you have access to a full trio or at least some family structure rather than just having a sequence of the patient. And one of the reasons for that is we can identify these de novo variants, which we only find a small number in each patient, but they have much higher prior of being disease-causing when you have unaffected parents and an affected child. The other explanation for that is recessive disease, like we just spoke about, but that's much higher when you have, say, related parents. Then you can kind of hypothesize that it will be a recessive cause. But again, as we just spoke about, you get that phasing information by doing the trio. So trios are incredibly valuable, but there are other family structures available as well. So you might have 2 affected children, and then you can have a quad. But even having anything other than just the single patient can be very valuable.
You've done quite a bit of creative work so far in your career. I was always very impressed with with the open reading frame work you were doing. Exactly like you said, this is something that nobody really actually was on anybody's radar, but there's actually something really interesting there. There's another one of your papers that I actually bring up a lot because I think it's another kind of non-intuitive but really creative example of genomics and drug discovery, which is some of the work you did as part of the NOMAD consortium on loss of functions in LRRK2. It'd be really great to hear from you about how that came about and some of the lessons you learned from doing that work. Because I think it's initially non-intuitive why it's so useful, but then I think once you dig into it, it certainly for me was a kind of aha moment about how genomic data sets could be used in drug discovery in really creative ways.
Yes, this project was a kind of typical science example of some of the big papers come from being in the right place at the right time. So this was a project that Daniel MacArthur had already initiated with 23andMe, where they were interested in kind of showing the value of humans as an experiment for drug discovery. So Parkinson's and LRRK2 is quite an interesting example here. So gain-of-function mutations in LRRK2 are known to cause Parkinson's disease. So because of this, there's huge interest in the pharmaceutical world of trying to develop inhibitors to LRRK2 as a treatment for Parkinson's. But when people had kind of modeled this in mice and other model organisms, they've seen some very severe lung, liver, and kidney phenotypes in the kind of knockout animals. But as we know, these animals are models, but they're not actually humans. But nature does this wonderful experiment where it introduces all this natural variation. And what can we use for that natural variation to look at what the effect of knocking down a specific drug target might be in humans? And there's a great companion paper to this as well by Eric Minakal that kind of looks at this on a more broad scale. And LRRK2 was just a specific example we were looking at. So what this allows us to do is to look at these loss of function mutations. So where we kind of predicted to lose half a copy of, half the amount of protein is what the downstream impact we expect. And look in kind of large scale biobanks or data sets with linked health information and say, well, what impact does that have in these people? So this was kind of a paper full of completely negative results, but that was exactly what we wanted. So I've got all of these flat line figures that were quite fun to create, but showing that there doesn't seem to be any increase in any lung, liver, kidney or other kind of severe phenotypes as far as we could test with the power that we had in humans that have half the amount of LRRK2 protein. So what this suggests is knocking down LRRK2 by half shouldn't be deleterious when it's done in clinical trials. So this was really kind of positive negative data, which is a really great thing to show.
Yeah, and I think as a general kind of class of research question, I got some advice very early on in my PhD, which was if you focus on a question or set of questions that no matter what the answer is, it's going to be interesting, then you'll be fine. Where you run into trouble is if you have a question that is only interesting if this one answer or the other. And then as a scientist, you're stuck with either getting the interesting answer or getting the non-interesting. And this is a perfect example of that, where if you'd found that people with a partial knockout of LRRK2 end up with significant kidney, liver, lung disease, then that's a really useful piece of information because you can tell the world, hey, we actually do need to be careful inhibiting this protein. And if you find the reverse, that actually we don't see much evidence that there are some people who have this knocked out their entire life in every cell of their body, like you said, by nature, and we don't see kidney disease showing up in their medical records or things like that, then it doesn't mean it's definitely safe, but it's a pretty good indicator. So it's a really good example of that principle.
Yeah, there's really important caveats. And one of those being that if you have it knocked down from birth, there are potentially response mechanisms in the body where they can kind of correct for it by maybe upregulating something else, which is not going to happen possibly if you give somebody a drug later in life. But as you say, it is kind of lifelong knockdown, whereas the therapeutic is only going to be later in life. So there's kind of it's not the same experiment, but it is kind of promising data.
Yeah, that is a really important and interesting caveat. Essentially, that there may be some kind of compensation where if you always have half the protein, then the body figures out a way to make it work. But like you say, if you introduce it instantly at some point later in life, you can't rely on that compensation to necessarily happen.
Yeah, exactly. And we also obviously can't predict off-target effect that the drug might have very easily.
And you recently, on the topic of advice to early career researchers, you recently started a research group. I'm really curious, how did it feel day one, walking into the new office? I'm sure it was just you on day one, but you had a plan to build the team. How did that feel? What brought you to that moment, and how's it been?
I guess one of the most disorientating things is I didn't walk into a new office. I stayed in exactly the same office in my own house. I started in September 2020, so very much having delayed from June 2020 because I didn't feel like starting a lab in the middle of a pandemic, which shows you a bit of our short-sightedness of how long we thought this thing would go on for.
Yes.
But yes, I started in September, and I think, gotten how long it was, I think it was nearly a year before I actually went into the office and found out how tall my team members were. So given I managed to delay for that amount of time, I managed to set up some recruitment. So actually, I only had a couple of weeks of being the only member of my team, and then very quickly had a DPhil student and a postdoc and actually an intern join the group. So that was quite nice. Science can be quite an isolating place anyway. You don't want to sit in your own house as the only member of your team for too long.
How has that transition been for you personally? What changed from being a postdoc, where you were obviously working as part of a team and had, often postdocs do work really closely with PhD students and others, but from making that transition then into really being able to set your own research agenda in a way you probably weren't able to before. What was that like, and what were you expecting, and how was it?
Yeah, no, it's great fun actually, and I learned to code during my PhD. I'm not going to say that I'm the best coder in the world, so it's great to be able to employ people who are better at coding than me to actually do my ideas. But I love kind of being able to create the environment that I want to work in, and making things kind of supportive and inclusive, and I really enjoy that side of things. The kind of key difference I think in the way that you work is as a student or a postdoc, you get to kind of say, oh, today I'm going to work on this, and you get to kind of shut everything else down for the day and just work on a sole bit of code, sole question for the entire day. As a PI, it's very much you're jumping about every half an hour and trying to reprogram your brain to kind of go from one task to about 20 different things that are on completely different topics. It's quite a challenge. I found that incredibly disorientating to start off with, but I think I've kind of got that one a little bit sorted by this point.
And what are you most excited about right now? What are the areas of research you and your team are working on? And also I'd love to hear about any areas that maybe aren't what you're working on, but as a whole, you think are exciting for the next coming years, that especially ones that maybe aren't on people's radar that you think in a few years will be saying, maybe like the open reading frames a couple of years ago when nobody was talking about it, but then it starts to it starts to become a lot more of a topic. What are you most interested in the group and in the industry as a whole?
We kind of have quite a broad focus in the group, from the very, very clinical stuff. So we've just put out a set of guidelines for how clinically we should interpret variants in the non-coding regions. So that was a really great collaboration with a lot of awesome people that think in the space. But also, as I said, we're trying to put a number on this, how many patients can we expect to diagnose through using the Genomics England data. But also we're trying to look more on the regulatory side of things and really understanding UTRs, hoping that will help us to interpret variants down the line. So there's a wonderful DPhil student, Nahama, who is looking at kind of how UTRs vary across different gene sets. So as we vary the tolerance to loss of function, we see massive differences in the makeup, the length and the makeup of UTRs. And also if we look at, say, dominant developmental disorder genes, they look completely different to kind of genes on a whole. So there's important things to learn about regulation, which hopefully downstream will help us interpret variants. And we've kind of got this big collaboration with Novo Nordisk, who are a Danish company, and Joe Hausen, who's the head of genetics, to look a bit more about UORFs and UTR variants and their role in more common phenotypes using a lot of UK biobank data. So really excited about that. Kind of on a whole, it's an interesting question. I think we're going to learn a lot from having the whole genomes in all of the UK biobank cohort. I'm really excited to see what people do with that data. I think there's going to be some really awesome things we can learn, but also some really cool computational and analytical approaches that people come up with how to kind of wrangle that data and learn new insights out of that. So I'm very excited to see where that takes us.
I completely agree. Do you think that on the topic of both whole genomes and large-scale data sets, and also understanding more about the non-coding regions, do you think there are enough people on the planet for us to actually understand these by looking at human data? And what I mean by that is, if we were to sequence whole genome, everyone on Earth, I think it's possible that the complexity in these regions is still too high, that we couldn't, using data from 6 billion people alone, really pick out which bases are important, which ones are not important, which ones cause disease. I know a lot of people agree with that statement, but a lot of people vehemently disagree with that statement. I'm curious where you fall on that, and then maybe we could talk about some of the other strategies to tackle that problem.
I think one of the important things is that you don't have to observe a variant to tell something about it. When you get to a certain number of people that we've sequenced, and when you look at variants that are at positions that are highly mutable, the fact that you don't see that variant is as informative as if you see it. So that implies that it's not compatible with life. So with variation that we already know is at saturation at the sample sizes we have now, so I'm talking about C to T changes at CG dinucleotides that are methylated, we know they have a very high mutation rate. And just by looking at those variants that you don't see, you can learn a hell of a lot. So I think the fact that humans have been under selection for however many number of years means you're not purely looking at a DNA sequence in isolation. You're looking at kind of what's happened in the past. And by combining, if we had 6 million people, the data, that would be amazing. I think we would be able to learn a hell of a lot from that.
Yeah, I completely agree. And I guess I need to update my numbers. It's probably 7 billion now, right? 6 billion is what it was when I was a kid. And that's the number that's burned into my mind, but no longer. Well, thank you. I think it's a great note to end on. I really appreciate you taking the time and talking about your work and being with us today.
Well, thank you very much. It's great to be here.
So if you want to follow Nicky and her work, she's on Twitter at Nicky Whiffin. You got the first name, last name, which is great. That means that you were a little bit early to the party. And I know
I just have a very obscure name.
Yes, that's right. I couldn't get the Patrick Short, unfortunately, too common. And I think you're hiring right now, right? So if people want to learn more about your group, what you do, you're Nicky Whiffin on Twitter, but I think your website as well is rarediseasegenomics.org. Maybe you could talk a little bit about who you're looking for and what they'd be working on.
Yeah, definitely. The best thing to do is to find the contact details through the website, or just by Googling me. Again, obscure name means I'm very Googleable. Slightly terrifying if I ever do anything wrong. But we're looking for kind of a senior postdoc to lead a collaborative project with Novo Nordisk that I briefly mentioned, looking at UTR variants and using kind of multi-omic within the UK Biobank. I think it should be a really awesome project.
Awesome. Well, great. Thanks, Nicky, and thanks everyone for listening. As always, we'd really appreciate if you could share this episode with a friend if you liked it, and leave a review on your favorite podcast player. This helps other people find us. So thanks very much for your time, and we'll see you next time.